Well, again, welcome everybody. And what I'm going to do first of all, before we get on to the CTG repeats, I'm just going to make sure that everybody's up to speed and we understand a little bit about the inheritance patterns of the disorder, myotonic dystrophy. So we know that myotonic dystrophy is an incredibly variable disorder. Individuals can be affected with the condition at a wide range of different ages, and the absolute severity and the presentation of symptoms is very different from one individual to another. And the CTG repeats are certainly important in that, but as we'll come back to later on, it's not the only thing that affects the absolute presentation of the disease in any one given individual. So regarding the inheritance of the disease, this is an inherited disease, so it's transmitted from one generation to the next, so it's passed on in the genetic material, the DNA. Now, myotonic dystrophy is what we describe as an autosomal dominant disorder, and what that really simply means is that it's not to do with the sex of the individual. So individuals of both sexes can develop the disease, and the disease can be passed on from parents of either sex. And the other important feature is that it only requires a single copy of the defective gene to be transmitted to an individual for them to develop the symptoms. So usually an individual only has one affected parent and the other individual is unaffected in the two parents. So what we're going to be seeing a little bit today, again, a few times today, are family trees. So I'm just going to first of all run through some of the things that we look for in family trees and how we interpret them. So over here on the screen, hopefully you can all see my mouse. I'm showing here, this is a square, and this is how we usually predict males or men in a family tree. And for each of the genes that we have in our genome, we have two copies of that gene, and below the square here depicting that this is a man, I've indicated those two genes here, these two little white shapes here. And for an unaffected individual, we usually indicate them as being unfilled. So this is the white square here, means this is an unaffected male. And because these are the two unaffected copies of the gene here, I've indicated those in white. Over here, I've indicated another male. It's square again, so it's another male, but this is an individual who's affected, so we've colored them in red to indicate that they are affected. And again, I've indicated the two genes that they have here, so they have this unaffected copy of the gene here and this mutated copy of the gene here that's giving them their symptoms. Over here we have an unaffected woman, so women, ladies are designated as circles, men as squares, ladies as circles. And again, this is an unaffected lady, so she's not filled in, and she has the two unfilled in, unaffected copies of the gene here. Whereas over here now we have a depiction of an unaffected lady, so she's filled in red, and she has one unaffected copy of the gene and one affected copy of the gene. So what would happen if this unaffected man now and this affected lady were to have children? What would be the possible outcomes in the children there? Well, all of the children would inherit one copy of the gene from their father and one copy of the gene from their mother. Now, because the man here, both of his copies are the unaffected copy, all of his children will inherit one of the white versions of the gene here, the unaffected version. Now, the mother, half the time she will transmit this unaffected copy of the gene to her children. So the half of the children that inherit the unaffected copy here will now have two unaffected copies, so they will be unaffected. But unfortunately, half of the children will inherit this mutant version of the gene here. And because they've inherited that, just a single copy of it, they will get the symptoms as well. And it works exactly the same for a male here who's an affected male with an unaffected female. Again, half of the children will inherit or all of the children will inherit one copy of the unaffected version of the gene from the mother, and half will inherit one copy of the unaffected gene from their father, but half will also inherit the mutant copy of the gene from their father. So the disease can be passed from either affected males or affected females to either affected sons or affected daughters. And that will happen, all of those things will happen in a 50% ratio. So on average, 50% of the children of an affected parent will also be affected. However, it's important to remember that's just an average. It doesn't mean that if a couple have 4 children, 2 of them will definitely be unaffected and 2 of them will be affected. It's an average across multiple families. So some families will be lucky and none of the children will inherit the mutation. Other families might be unlucky and more than 50% of the children might inherit the mutation. So again, just to clarify one other thing about the inheritance. So on this side of the family, where this daughter has inherited the mutation, that was a 50% risk from her mother that she inherited. She unfortunately did inherit it. When she transmits it again, it will be a 50% risk that she will pass that on to her offspring down here. However, on the other side of the family over here, where this male didn't inherit the mutation, and as long as his partner is unaffected as well, then their children will be at 0% risk of inheriting the disease. So for any individual that doesn't inherit the mutation, their children are essentially at 0% risk. All those who do inherit the mutation, their children will be at 50% risk. So that's the very simple genetics, is that the individual has a 50% chance of transmitting the disease to the next generation. However, as many of you will be aware, in myotonic dystrophy, it's not quite so simple, in that the severity of the disease, as we've said, is extremely variable, but that's not random. That follows a distinct pattern where in most families, the symptoms become worse in successive generations. I've illustrated that here on this family tree, where often the individual in the first generation only has a late onset form of the disease. Their children might have the adult onset form of the disease, but often within 2 or 3 generations, we end up with a very severe form of the disease in the 3rd generation, where it might be the juvenile onset of the disease or even the congenital onset, where the symptoms occur at birth. So this is a clinical phenomenon known as anticipation, where the disease gets worse from one generation to the next, and the symptoms become, the age of onset becomes progressively earlier in successive generations. And we'll come back to why that is in a minute. Again, although the disease can be transmitted from parents of either sex, so you can inherit the disease from either your father or your mother, the absolute severity of the disease can differ depending on whether the mutation was inherited from the affected father or an affected mother. And what we know is that most of the children born in the later generations who have the severe congenital form of the disease, the majority of those have an affected mother. There are a few that have an affected father, but the majority have an affected mother. Whereas if we go back to the generation before that, we find that many of the individuals who transmit the disease when it progresses from the late onset form to the adult onset form, this is usually the father here. So relative to the congenitally affected children in this generation, there's very often an affected grandparent. But that effect is not as strong as the excess of transmitting mothers in the middle generation here. So we talked a little bit about the inheritance of the mutation. So how is the information transmitted from one generation to the next, and what is the nature of the mutation? So the genetic material that's transmitted in the egg and the sperm is called DNA, deoxyribose nucleic acid, and the structure of this molecule was determined a little over 60 years ago now, in 1953, by James Watson and Francis Crick. And what they discovered was that the structure of DNA was this double helical structure here. It's also illustrated by this model in the middle here, but this is a cartoon of it here as well. So as well as being a very beautiful structure, what they also discovered that it was made up of 4 very simple chemical letters. So DNA is a very long molecule, and what it essentially is, it's a string of 4 very simple chemical letters. So those chemical letters are A, C, T, and G. And if we were to go in and look at a piece of DNA, it would essentially look like a random collection of A's, C's, T's, and G's. Turns out that that actually is a very complex code that contains all of the information necessary to build all of the individual proteins, to assemble cells, and to assemble intact human individuals, all in that code of A's, C's, T's, and G's. Some of that code we understand, but a lot of it we're still understanding exactly how that code is translated into cells and proteins and intact individuals. Nonetheless, what we know is that there are some regions where the code appears to be not random, but it appears to have some simple patterns. So for instance, the sequence CTG here is repeated a number of times. So we can see there's a CTG, CTG, CTG repeated a number of times. And we find that that type of region is quite common in the human genome. There are regions where a simple sequence is repeated a number of times. What we find is very often when we have a sequence such as that, the number of copies of the CTG repeat varies from one individual to another. So one individual might have 5 copies, another individual might have 10 copies or 20 copies. And this type of variation is actually the variation that's used in DNA profiling, that's used to match, for instance, scene of crime samples to suspects, and it's also used to establish relationships, for instance, in paternity testing, etc. So that type of variation is very common, but usually it doesn't matter to an individual whether they have 5 repeats or 10 repeats or 20 repeats. It makes no difference to their function. But we know now that a number of genetic disorders are associated with changes in the number of such simple sequence repeats, where it does matter very much exactly how many repeats an individual inherits. And myotonic dystrophy type 1 is an example of such a disorder. So the mutation that is associated with myotonic dystrophy type 1 is a variation in the number of copies of a CTG repeat in a gene in our genome called DMPK. So everybody in the population has 2 copies of this gene, but the number of copies of repeats in that gene vary very often from one individual to another. So many individuals will have 5 copies of that CTG, but it varies in unaffected individuals. 10, 12, 13 copies are very common. 20 repeats is a bit less common, but still out there at relatively high frequency in the unaffected population. Again, 30 is a bit rarer in the population. Maybe 1 or 2% of individuals have 30 repeats. 40 repeats are again much rarer again, so maybe less than 1% of the population have 40 repeats. If the number of repeats, however, increases up to 50, then it become associated with symptoms. So some very minimally affected individuals with myotonic dystrophy will have 50 repeats. And 60 and 70 repeats again would be associated with very mild late onset symptoms. But as we get up into the 80s and 90s and 100s, then the symptoms can start to become more severe, can be associated with the adult onset form of the disease. So many individuals with the adult onset form of disease would inherit 200 or 300 copies of the repeats. But again, as the number of repeats increases, the severity of the symptoms increase and the age of onset decreases. So in the later generations, we find that some of those individuals with the congenital form of the disease, where the symptoms start at birth, may have inherited many hundreds or even 1,000 or more copies of the repeat. So one of the first things that we found when the mutation underlying myotonic dystrophy was discovered was that a large part of the explanation for why the symptoms might be so variable between individuals was related to the number of CTG repeats that an individual inherited. Individuals who inherited relatively small numbers of repeats would have a mild late onset form of the disease, whereas other individuals who inherited much larger numbers of CTGs would have more severe symptoms and an earlier age of onset. And again, what we also found was that repeat expansion provided some explanation for the anticipation that we see in families. So the normal copy of repeats, so in range of 5, 10, 20 repeats, that's usually stably transmitted from one generation to the next. So if a parent has 10 repeats, their children will very likely also inherit 10 repeats. Whereas once an individual has more than 50 repeats, it becomes genetically unstable, and it very often changes then when it's transmitted from one generation to the next. So for instance, in this family here, we have a man who's inherited 72 repeats. Associated with that is a relatively mild late onset form of the disease. But when he transmits that to his children here, and I'm showing a much simpler version of the family tree here, so I haven't got his unaffected partner and I'm not showing any of the unaffected children, so 50% of his children won't have inherited this copy of the gene, they'll have inherited the other copy and they'll be unaffected. But those that do inherit the mutant version of the gene, nearly always the number of repeats is different. So here it was 72, his children have inherited different numbers of repeats, but not only are they different than each other, different than was present in the father, there nearly always increases in number. And because the number of repeats is increased, their severity is greater and their age of onset is earlier. Again, that happens again in the next generation, such that in the next generation we now have many hundreds of repeats. And again, this can be associated either with the juvenile form or the most severe congenital form of disease when we have many thousands, or we can have many thousands of copies of the repeat. So this increasing number of repeats from one generation to the next is what explains the anticipation. The symptoms become progressively worse and successively earlier in succeeding generations. What this also provides a partial explanation for is the sex of parent effects as well. So we know that these relatively small expansions here, where we've got maybe 50, 60, or 70 repeats, that's particularly unstable when it's transmitted by a male. So that nearly always increases up into the hundreds of repeats. Whereas when a female has maybe 50 or 60 or 70 repeats, when she passes that on, it will get a little bit bigger, but not that much bigger. In the next generation here, many of these men are infertile, so that's why we don't see so many men transmitting the disease into the next generation here. And once the repeat gets up into the hundreds, then it becomes very unstable when transmitted by a female, and again, often expands into the high hundreds associated with the congenital form of the disease. So we know that there's a correlation between the number of repeats that an individual has and the severity of the symptoms, and that's illustrated on this graph here, where on the axis along the bottom here we have the number of CTG repeats, and this is the number of CTG repeats that were measured in the diagnostic test. And on the axis, the y-axis up here, we have the age of onset of that individual. And this line here represents the correlation, so the association between the number of repeats and the age of onset. And what this shows us is this line goes down here, so the more repeats an individual has, the earlier their age of onset. And that's a very strong correlation here. However, what you'll notice is that not all of the individuals here are on that line. There's a lot of variation. So that means that although on average, the more repeats an individual has, the more severe their symptoms and the earlier their age of onset, the actual predictive value of the number of CTG repeats is relatively low. So what that means is that a lot of error associated with predicting exactly the disease severity associated with the number of repeats. So essentially, although that white line is the average response, the kind of the pink shape here now dictates the boundaries of that. So we can kind of illustrate what the value of this is. If we think about someone with 500 repeats, then on average, they would have an age of onset and around the mid-20s here associated with that. However, as I've said, there's a lot of error associated with that prediction. So actually the symptoms could be much less and they might not get them until their late 50s, or the symptoms might actually be much earlier and they might get the juvenile form of the disease associated with this measured number of 500 repeats. As I come back to later on, that's why anyone should interpret their own number of repeats with extreme caution because of the variability associated with predicting exactly how severe the disease will be based simply on this measure of the number of CTG repeats that's provided in the standard diagnostic test. So what is the reason that the number of repeats isn't more predictive? Well, some of that is due to natural variation. So individuals differ in their response to having the myotonic dystrophy mutation for a whole bunch of reasons. Those can include interactions with the environment, but also the rest of the genetic variation in the genome. So for exactly the same reasons that everyone in the unaffected population is different from one another, everyone in the affected population is also different from one another in all sorts of different things over and above the fact that they might have the mutation associated with myotonic dystrophy. But also one of the other factors that we now know is important is that actually the number of repeats that we measure or that's present in an individual isn't stable. So in just the same way that the number of repeats can vary from one generation to the next, we now know that the number of repeats can also vary throughout the lifetime of an individual. And this slide here shows some data that's generated in my lab where we're measuring the number of repeats in individual cells in the DNA from an individual with myotonic dystrophy. And we've done this test on that person at 2 different time points. So first of all, when they were 36 years old, and later when they were 49 years old. And essentially what we're doing here is we're analyzing the DNA in individual cells. So each one of these bands here, this dark band on the autoradiograph, is a measure of how long the DNA was in that cell. So we've looked at 100 or so cells here, and we've resolved them such that the fragments of DNA containing less repeats are down the bottom of the picture here. Those fragments containing more CTG repeats are towards the top here. And this scale here is the number of CTG repeats. So this cell had around about 250 CTG repeats in it. This one here had about 500. And you'll see that on average, probably here, the number of repeats in 36 years was probably somewhere around about 200 repeats. Whereas when that same individual was sampled several years later, we see now that there are many more cells containing 300, 400, 500, now 700, 800, some probably up to 900 or so repeats. So clearly the average number of repeats has increased during this 13-year period when the blood sample was taken. What you'll also notice, though, that this boundary down here, this lower end of the distribution, is pretty similar in both samples. And what we think is happening is that this lower boundary here probably represents how many repeats that individual inherited from their affected parent, and that's what's happened during the lifetime of this individual is that the DNA has continued to expand, gradually getting bigger in different cells as the individual ages. And again, what's important to remember is the diagnostic test measures the average number of repeats. So if we'd done the diagnostic test at this point, we'd have probably measured around about 200 repeats. If we'd done the diagnostic test at this point, we'd have probably measured around about 300 repeats. So the result of the diagnostic test very much depends on how old the individual was when they did the test, as well as how many repeats they inherited from their affected parent. And what we've shown is in a research setting where we can use this very sophisticated test to try and estimate how many repeats an individual inherited. When we do that, we're much more accurately able to predict the relative severity of the disease. Much more accurately predict the age of onset of symptoms. However, still, even using that method, there's still variation between individuals. So 2 individuals inheriting the same number of repeats still won't have exactly the same age of onset. There's still quite a lot of variation. And again, some of that relates to variation in other genes and environmental factors, and some of that relates to the rate at which the repeat gets bigger. So we've also shown that individuals who inherit a similar number of repeats, how quickly that then gets bigger through their life will also impact on the disease. So those individuals in who the repeat gets bigger more quickly throughout their life tend to get the symptoms earlier than those individuals who expand the repeat more slowly. Again, why some individuals expand the repeat more rapidly than others, again, is something we're only just starting to investigate, but is likely, again, due to variation in other genes in the genome associated with DNA repair and DNA metabolism. This slide here, this movie, shows a simulation of what we think is happening to the number of repeats in an individual. Again, each little black dot here is representing the number of repeats in an individual cell in the body, and as you can see, as this individual ages, on average, the number of repeats increases. And this is a useful way of us understanding how we end up seeing the different numbers of repeats in different cells when we sample an individual at a given time point. And it also gives us a way of trying to predict what the number of repeats that was inherited in an individual was. So in our experiments, we would measure the end point of this distribution at a given point in time. What we can then do on the computer is essentially run this backwards to try and predict how many repeats an individual inherited, and again, understand what the relationship between that inherited number of repeats is and disease severity is. And as I said earlier on, that gives us a much better indication of disease severity. What we also know is that the number of repeats as well as changing throughout the lifetime of the individual in blood, which is obviously the tissue that's usually mostly used for the diagnostic test. We know that a similar thing occurs in other tissues, and actually that very often happens much more rapidly in other tissues. So now we're looking at blood and muscle from the same individual sampled at the same time point. This is their unaffected copy of the gene down here, so that doesn't change at all over time or between tissues. But you'll see here that there are more of these large bands in here containing more repeats in the muscle cells than there are in the blood cells. So the number of repeats in muscle cells is typically much larger than it is in blood cells. So what we think is happening in patients is that an individual inherits a given number of repeats, and that's relatively stable during embryogenesis. So for instance, this individual here inherited 300 repeats from their parents, and at birth there'll maybe be 300 repeats in the muscle cells and 300 repeats in the blood cells. 300 repeats in the muscle cells might be associated with actually very good muscle strength, as illustrated on this graph over here, where the height of this graph illustrates good muscle strength. But as that individual ages, the number of repeats starts to change. That changes more rapidly in muscle cells than it does in blood cells. And as the number of repeats increases, that then has more of an impact on the muscle cells and the function of the muscle. So the muscle may start to atrophy and muscle strength may decrease. And as the repeat number increases as the individual gets older, that becomes more and more pronounced. So we think that the changing number of repeats is driving both the progressive nature of the symptoms, why the symptoms become worse, and also telling us why certain tissues are affected more than other tissues. So the repeat increases relatively slowly in blood, so we think that's why there aren't many major blood associated symptoms, but it happens very rapidly in muscle, and that's why we think there are many major muscle associated symptoms in myotonic dystrophy. So that's a bit of the background there to how the number of repeats is changing and what we think the consequences of that are for individuals. What we've also discovered over the last few years is that, again, the story is not even as simple as that. So although the majority of individuals with myotonic dystrophy have an expansion of this CTG sequence, and that's illustrated here in this now schematic representation of the gene where each one of these little bars is a copy of the CTG repeat, and I've shown here the type of mutation that we get in most typical myotonic dystrophy families, where there's just an increase in the number of red bars. So there may be probably 50 or so here, and there can be many hundreds illustrated here by this kind of gap here, where I'm just illustrating that there are lots of CTG repeats. What we now know is that there are a subset of individuals with myotonic dystrophy where they have an additional type of mutation. So as well as having lots of CTG repeats, they have some other sequences that are also represented within the CTG array. And this illustrates one pattern of what we call variant repeats, where it's not just CTG repeats. In this case, it's CCG triplets in here, and also a few CGG triplets in here, and another region here where we see CTG followed by CCG a number of times. And we actually first discovered this pattern of mutation in an unusual family in which the symptoms of myotonic dystrophy were actually slightly milder than is present in a typical family, but that particular family also had some additional symptoms that were not present in your average myotonic dystrophy family. So it looks like we've now discovered similar types of effects in around about 5% of individuals with myotonic dystrophy. And it looks like the general story is that individuals who have these atypical mutations tend to have slightly milder forms of the disease. I'm going to just illustrate one example of a family that we identified here that's very unusual because it shows reverse anticipation. So rather than the number of repeats increasing in successive generations and the symptoms becoming worse, in this family, the number of repeats was observed to decrease and the symptoms to become less severe. So the reverse of anticipation. What we now know is that the sequence in this family, again, is not the pure CTG repeat, but again, this family has some of these variant repeats, these interruptions within the CTG array, and that starts off relatively few in the first generation here, but by the time we get down to this third generation, there are hardly any pure CTG repeats left. It's now almost completely replaced with this mixed type of sequence. And again, the general picture that's emerging is that individuals who have these alternative structures tend to have less severe symptoms, and we can often see the number of repeats decreasing in successive generations. However, again, I should point out that doesn't always happen. We have other families where the number of repeats does increase, and we do see the anticipation that we see in the typical families that just have the pure CTG repeats. So then a quick summary of what I've told you so far. The number of CTG repeats is correlated with disease severity. So the more repeats an individual has, the worse their symptoms and the earlier the age of onset. The number of CTG repeats grows in size throughout life. So whatever you inherit, that number then continues to increase throughout the lifetime of the individual. And that happens bigger in the affected tissues such as muscle than it does in some of the unaffected tissues such as blood. And again, how quickly that happens also modifies disease severity. So those individuals in which the repeat gets bigger more quickly tend to, again, have more severe symptoms. And again, in a subset of individuals, they have these variant repeats that, again, it's not just pure CTGs, but there are other sequences mixed up into the mutation, and that can modify the disease severity, generally making the disease milder, and also can change the risk of the relative disease severity of an affected child. So it doesn't change that 50% risk that the child will inherit the mutation, but it just changes the relative risk that the disease will become worse in successive generations, tends to reduce that a bit, but not eliminate it. So everything I've told you about so far is associated with myotonic dystrophy type 1, and that's the most common type of myotonic dystrophy in most of the populations. But there is another type of myotonic dystrophy type 2, and the genetics of that are similar, although generally less well understood. So myotonic dystrophy type 2, again, only requires a single copy of the defective gene, and again, can be acquired by individuals of both sexes and can be inherited from individuals of either sex. So again, fathers can pass it to sons and daughters, and women can pass it to their sons and daughters too, and both are equally affected. And it turns out that this is also caused by an expansion of a repeated sequence, but rather than being 3 letters this time, so it's not CTG, this time it's 4 letters CCTG, and in a different gene, and that gene is officially called CNBP, although sometimes it's called ZNF9. And the number of repeats, again, is usually less than about 30 or 40 in unaffected individuals, and is from 50 up to many thousands of copies of the repeats in affected individuals. And in this case, the number of repeats is even more unstable. It changes even more rapidly throughout the lifetime of the individual, and is probably also changing rapidly from one generation to the next, but actually because it's so unstable in the blood, it's actually very difficult to measure what's happening from one generation to the next. And overall, the correlation between the number of CTG repeats and disease severity in myotonic dystrophy type 2 is even less clear than it is in myotonic dystrophy type 1, although there's probably a similar overall relationship. So that then just leaves me in the last few minutes to just have a quick look at a couple of the different types of genetic diagnostic results that some individuals may have received. And there's obviously a lot of jargon on there, and let's just have a look at this one here from the Baylor College of Medicine and see if we can interpret what are the key things that an individual might need to take from this. So first of all, they tell you that they're testing for the myotonic dystrophy mutation, and that's usually done on the basis that an individual has a family history of the disease and or a clinical diagnosis, and the molecular test is usually done to confirm that this really is a case of myotonic dystrophy. And they're looking for what's called here a trinucleotide repeat expansion. So a trinucleotide is essentially referring to the 3 bases, those CTGs, that are expanded in individuals with myotonic dystrophy. Here they've called the gene myotonin protein kinase, Again, that's not the official name of the gene. Officially, the gene is called Dystrophy Myotonica protein kinase, DMPK. So sometimes you might see myotonin protein kinase. Other labs may report it as DMPK or Dystrophy Myotonica protein kinase. So the DNA has been taken from this individual. We've anonymized this report here, and it's used to study the size of the CTG repeat in the blood DNA of that given individual. And what they're reporting here is that one expanded allele, so the allele is another name, is the technical name that we give for the different versions of the gene that an individual has. So each individual will have two versions of the DMPK gene, one inherited from their affected parent and one inherited from their unaffected parent. So we'll have two alleles, two different copies of the gene. And what they're saying here is that on the mutated version of the gene, there are 800 repeats, or somewhere between 800 and 1,000, 1,100. And again, this is very often with this particular test, it just measures the average number of repeats, and there's not a nice discrete number of repeats that's measured. It's just an average number, and it's a bit blurry. And that's because the number of repeats is varying from one cell to the next. And they're saying on the other allele, is within what they call the normal range. So essentially what they mean by the normal range is the range that's seen in unaffected individuals. So we would assume that that's probably in the region of 5 to 20 repeats. Might be 30 repeats, but probably in the range of 5 to 20 repeats. So what they're saying is this results indicate that this individual has the common trinucleotide repeat expansion mutation observed in the majority of patients affected with myotonic dystrophy. So they're saying this is a positive diagnosis, and it's a molecular confirmation of myotonic dystrophy. Again, it doesn't necessarily mean that that individual will necessarily have symptoms at that time. So again, if this is particularly an individual who is maybe being tested because another individual in the family has the symptoms, it's possible that when they have that test, they're actually asymptomatic. So again, it's possible to get a positive molecular test before the symptoms are actually apparent. But if an individual has a positive molecular test, it's very likely that at some point in their life they will develop the symptoms. Again, predicting exactly at what age the symptoms will develop is, like I've said, is we can predict that to an extent, but not 100% accurately. Then it's saying that again, that if someone receives a positive molecular diagnosis, it's likely that other people in the family will also carry the mutation, and they would be advised to have genetic counseling and a DNA test to confirm whether or not they are at risk. Down here, the methodology, Southern analysis, NCO1, BAMH1, PCR, etc. These are all technical laboratory details on exactly how the test is done. It's not really necessary for you to understand exactly what this means, because it's just the labs are doing the test in slightly different ways from one lab to another. Again, here they're telling us again what they consider, the normal range here, 5 to 35, and mutations being greater than 50, they would say as being a positive diagnosis. And then we have this kind of gray zone in the middle here, 36 to 49. So individuals with this number of repeats would be unlikely to have any symptoms, but their children might be at risk. So someone with 45 repeats, for instance, wouldn't have any symptoms, but they might transmit an allele of, say, 50 or 60 repeats to their children, who would then be at risk of having a mild form of the disease. Then again here, there's the small print, and as you can see, it's very small, and this is again just saying that mistakes do happen. The people running the tests are humans, and they can make mistakes too, so the samples could be mixed up, or they could have done the test incorrectly. Again, that happens very rarely. They're very, very careful in these diagnostic labs. They have lots of procedures in place to make sure that mix-ups and mistakes don't happen, but again, they are human too, so very, very occasionally there can be a mistake, but that's very, very rare. They do these tests very, very accurately, by and large. So that's a report from Baylor College of Medicine. We have another report here from the Regional Molecular Genetics Lab in California. And again, it records much the same information. They're saying that they're testing for myotonic dystrophy, and that there's already a family history, and that this is a test to confirm using the molecular approach. And again, they're using genomic DNA, so that just means the DNA that's in the nucleus of the cell. So there's not the mitochondrial DNA that's passed on just from mothers. This is the genomic DNA that's inherited, 2 copies, one from your father, 1 copy from your mother, and they're looking at that in blood. And they've used, again, PCR and Southern blots. Again, these are the technical methods used for measuring the number of CTG repeats within the myotonin protein kinase gene. And again, they're showing here that on one version of the gene there's 505 CTG repeats, so that's a positive molecular diagnosis, and on the other copy there's a size of 4 repeats, which would be the unaffected copy inherited from the unaffected parent. So again, much the same information as we saw on the previous one. Again, here, this slide here is just, again, you've got some of those definitions of the repeats that we just talked about on the last 2 examples there. And again, some more definitions there. And again, what we consider to be the approximate sizes. So less than 40 repeats is unaffected, 40 to 50 is this intermediate zone, unaffected but risk of affected children, and greater than 50 is a positive diagnosis. And all these other terms down here, again, those are associated with a specific method, and you don't really need to worry about those. So, interpreting the genetic test result, so very likely, as well as a positive molecular diagnosis, you will have heard a specific number of repeats associated with that test. But again, it's important to remember that that's the average number of repeats. It's quite approximate, so the test used only gives you a rough idea of the number of repeats. And again, it only tells you the number of repeats at one point in time. And as we've already said, the number of repeats is changing throughout the lifetime of the individual. So the number of repeats that will be reported on the diagnostic test depends on how old that individual is. So again, without taking that into account, it's very difficult to really interpret too much from the number of repeats. And again, another important factor is that the diagnostic test does not detect the presence of those variant repeats. So again, those are relatively rare, probably less than 5% of individuals have that slightly different type of mutation, but nonetheless, the standard diagnostic test doesn't tell you one way or the other whether an individual does have those variant repeats. So overall, although many individuals are interested in knowing the number of repeats that they have, they might be interested in comparing that to other individuals in their family, looking at what's happening from one generation to the next. As I've said, because of these caveats, that it's just an average at a single time point, you should not overinterpret that number. So just because you've got a high number or a low number, you shouldn't take that as a definitive guide to exactly how severe an individual's symptoms will be. There is a correlation there, but again, it's a very approximate correlation. So the number of CTG repeats is important in defining disease severity, but, and it's a big but, that's why I put it in big letters here, there is variation in other genes that also contributes to the severity of the disease. And again, we're just at the early stages of trying to understand what the impact is of variation in other genes. But again, lifestyle, life experiences, and the environment are also important in defining exactly how severe the symptoms will be in a given individual. And again, we don't really know what many of those factors are, and again, we're just starting to understand those. Again, it's important to remember that everyone with myotonic dystrophy, just as everyone who doesn't have myotonic dystrophy, those are unique individuals. So they might have their own unique mutation, but they will also have a unique set of other genes. They'll have had a unique lifestyle, unique set of life experiences. So each individual will be unique, and you shouldn't feel that anyone shouldn't feel that they're being defined by the number of CTG repeats. So CTG repeats are important, but they're not the only thing that's important for an individual. So I'll leave it there, and I'll be happy to take any questions. Leslie, if we have any questions?
Yes, we do. Thank you, by the way.
Great. So hopefully that was informative.
So there's a question. Is it possible to get a false negative reading?
Yes, so that is possible. It happens relatively rarely, and there are 2 primary reasons for that. So one is there's some sort of mistake in the lab. That's pretty rare. So as I've said, the labs do these tests very, very carefully. But we know now that there are a few cases of negative results that are false negatives, and that's associated with some of those individuals who have those variant repeats where the sequence is not just the CTG repeat. So some of the tests that are done assume that it's going to be a CTG repeat, and if the sequence is different, then the test may not work. Again, that's relatively rare, but it has happened. Usually what happens is if there is a strong family history and there is a good clinical diagnosis so that the clinical team is pretty convinced this really is Marginal Entry type 1, then they may repeat the test and try it in a slightly different way to see if they can overcome this potential problem that can happen. So it can happen, but it's relatively rare.
The next question is related to DM2. Are the repeats associated with someone with DM2, are females also more likely to hold a stronger onset to the next generation?
So yeah, so in Marginal Entry type 2, the anticipation that we see in Marginal Entry type 1 isn't really obvious, and it's debatable whether it really exists or not. So although individuals vary in their symptoms, the amount of variation isn't as great as we see in Marginal Entry type 1, and then this anticipation isn't as obvious, and consequently there is no congenital form of the disease, so there is no obvious sex of parent effects in terms of the number of repeats that are transmitted.
There were a couple questions related to family members. Could they have different types? One, I believe, mother said a son has been diagnosed with DM1. Would the other son have, who does have myotonic dystrophy, would it be type 1? And then someone asked, I guess DM2, someone has DM2, I'm assuming a mother or father, and could the child have DM1? Is there that kind of
no. So that isn't possible. It would be, so both are rare disorders. They are caused by mutations in different genes. So what is possible, but I don't know if there are any examples, and if they are, there will be one or two in the whole world where an individual or a family could have both mutations in the same family, but that would just be pure coincidence, but it could theoretically happen. But if the family has a Myotonic dystrophy type 1 mutation, it cannot change to Myotonic dystrophy type 2, or if someone has Myotonic dystrophy type 2, it cannot change to Myotonic dystrophy type 1. So extremely unlikely that that both would be present in the same family, although that's theoretically possible. But if you've got a diagnosis of Myotonic dystrophy type 1 in one individual and another individual in that family also has Myotonic dystrophy, it's very likely to be exactly the same type. Although again, different numbers of repeats and other genetic variation could mean that the symptoms could still be quite variable between those individuals. And I'm sure that's apparent in the majority of families, that the absolute severity, the range of symptoms is very different from one individual to another, even within the same family.
Someone asks if there is a type 3.
Not that we know of. So the vast majority of individuals in most populations have type 1. There are a few populations where type 2, so some parts of Northern Europe, in Germany and Finland, type 2 is quite common. Some areas of the States it's a bit more common. But we don't know yet of any families that clearly have myotonic dystrophy symptoms and don't have either of those two mutations. So again, there have been occasional reports of families that have symptoms that are kind of a bit like myotonic dystrophy, but I think on closer examination they've turned out to not really have the characteristic features that we would use to define the disease as myotonic dystrophy.
Okay, here's a question. Is the variant repeats issue going to affect how likely the current treatment ideas work in these different patients?
Yes, so that's certainly a possibility. So one of the main targets is this. Well, it's not the CTG repeat at the DNA, but in the intermediate molecule between the DNA and the protein is another molecule called RNA. And in the The DNA, it's a CTG repeat. In RNA, the code is slightly different, so they don't use the letter T. We use the letter U in RNA, so there's a CUG repeat in the RNA. And it's thought that most of the symptoms are to do with that RNA doing things that it shouldn't do. So one of the main targets now is to destroy that CUG RNA. And one of the ways they're doing that is using a complementary sequence that will bind to that RNA and stop it doing the bad things. And again, that one of those treatments that is using these so-called oligonucleotides is based on the assumption that the mutation is a CTG in the DNA and it will be a CUG in the RNA. So individuals who have variant repeats may respond differently to that drug than an individual who has a pure CTG repeat. However, as I've said, their symptoms are generally milder. So again, they may not need the, well, I guess that's not a good way, but anyway, so they may not actually be in the clinical trial in the first place, depending on how mild their symptoms are. But again, it's certainly one thing that we have to consider going forward. The other way, obviously, to look at that is because their symptoms are milder, it's actually telling us something about how the disease is caused. So again, it's giving us new clues to how we could treat the disease as well. So one of the things we're thinking about, it's going to be a long time before we can turn that into a treatment, but is the idea that if we could make variant repeats in individuals who have pure repeats, that would decrease the severity of their symptoms, we think. So again, it's giving us ideas as well as potentially confounding some of the current ideas or approaches.
Here's a specific case where someone is talking about the father of two sons. The father's symptoms are very mild, but both sons have it severely. Yeah. And they question whether the mother could also be affected.
And again, it's possible, but it's very unlikely. Again, so myotonic dystrophy is a rare disease, so the chances of two affected individuals becoming partners is relatively low. So although I said, so the congenital form of the disease with the symptoms present at birth, that's relatively rare to be transmitted from a father, but there are examples of that, so it is possible. What is reasonably common, though, is for an affected father to have children with the juvenile form of the disease, so not symptoms at birth, but symptoms starting nonetheless within the first decade of life. So maybe slow development at school or some physical problems during adolescence, etc. That's the juvenile form of disease can be transmitted relatively frequently by affected fathers, but usually not the congenital form with onset at birth.
Mm-hmm. Okay, I have two more questions, and I just want to let audience members know that if your question is very specific and asking about sort of a physician's advice, I'm not going to pose that to Dr. Moncton because he is a geneticist. Geneticist, did I say that right?
Yeah.
And you probably don't want to answer those kind of questions.
Yes, yeah. So actually, I should point out I am not medically qualified. So I'm a PhD scientist, but I'm not medically qualified. And again, I think you gave all the warnings at the beginning that I'm not giving anybody any specific medical advice.
Right. Okay, so last two questions. Is there any case where the number of CTG repeat decreases as a person ages? And I think you mentioned that.
Well, so the average number of repeats always increases. We've not found anybody where the number of repeats through their life decreases, okay? Not on average. We see in a few cells, so in some of the cells it may get a bit smaller, but not enough, we don't think, to impact the severity of the symptoms. But you're right, from one generation to the next, we do see occasional decreases in the number of repeats. But through the lifetime of an individual, we don't generally see that. Again, we've not got any good examples where that happens in all of the cells. It happens in a few cells, but not many, and not enough, we think, to impact the course of the disease. But again, in the long run, that's one of the things that we're trying to be able to do. If we can come up with drugs that stop the repeat getting bigger and actually make it get smaller, then again, we think that would be therapeutically very beneficial. But again, that's very early stages of thinking about how we could try and do that or develop a drug that would have that effect.
And a really good transition here for the final question. Are any of these treatments currently available, or any treatments currently available?
Well, so there are some treatments available. No, there are no cures, as obviously everybody knows, and there are no real treatments yet based on our molecular understanding of the disease. But there are a number of things, number of treatments that can be used for specific symptoms. Again, I'm not really the best person to address that, but you'll be familiar that something like modafinil can be effective in some individuals for addressing issues with sleepiness, and I can't remember the name of the drug now, but there is a drug that in some individuals is effective for the myotonia, the muscle stiffness. And obviously there's the heart defects that again that can be monitored very carefully, and pacemakers might be effective in reducing some of the arrhythmia. But as I've said, there have been some developments in terms of our understanding, as like I said, over the maybe this year or hopefully if not this year, early next year, there will be a trial using these antisense oligonucleotides, like I said, that target the RNA, and we would expect in the majority of patients that will be effective. Again, we know from animal models, the hope is that that will be effective in humans as well. Yeah, so, and like I say, we are also, and that's one of the things that I'm going to be talking about at the MDF meeting in Washington. So we're just starting a new trial. We just started a few months ago a new trial in Europe to test the effect of exercise and cognitive behavioral therapy on minimizing the impact of the symptoms. So again, we think, everyone believes, that moderate exercise should be good for individuals with myotonic dystrophy. Just like it is good for everybody in the general population, it should be good for people with myotonic dystrophy as well. But we don't really have any hard evidence for that. So that's one of the trials that we've just started here in Europe to see whether exercise therapy really does have an impact on an individual's quality of life. We believe it should, but we really need to get the data to prove it.
Well, that sounds like a very exciting study. Is it just
Yes,
in Europe or any U.S. locations?
No, at the moment it's just in Europe. It's in four countries in Europe. It's funded by the European Union. So there's a trial site in Newcastle, in the UK, Paris, in France, Munich in Germany, and Nijmegen in Holland. And that's being coordinated by Basile van Engelen in Nijmegen as the coordinator of that project. And my group, we're not doing any of the patient work here, but we're doing some of the genetic analysis. So again, we will be doing those sophisticated genetic tests that go beyond the standard diagnostic test. We'll be doing that in this cohort of patients to see if that gives us any clues as to why some individuals are more severely affected, or why they may or may not respond to the exercise therapy in the same way as some of the other patients.
And when do you anticipate the results will be available?
So the patients are just starting to be recruited now. So the whole course of the experiment takes about 10 months. And obviously we're going to be recruiting probably over the next year or so. So it's going to be a couple of years before we really start to get a good indication of whether the intervention has worked or not. But we're excited about that, and we're very positive that it will have an impact. Thank you. Thanks again for inviting me and giving me the chance to talk to you all this evening. Thank you.
Thank you, and thank you very much for your participation.